So we're now going to get into the biodiversity part and start going through different groups of organisms. So our domains of life are broken into three domains: our Eubacteria, Archaea, and Eukaryota. So two of those domains used to be referred to just as prokaryotes. Okay, so pro meaning before, and then karyo, I guess I should swap this out, karyo meaning nucleus or nuclear. So referring to the nucleus and essentially saying that these groups don't have a nucleus. So usually that's what that means, no nucleus. What it means in a larger sense is this: the nucleus contains the genetic material. The entire structure is a membrane, a phospholipid bilayer, and generally in our cells it's actually a double membrane, so there's two phospholipid bilayers, and then inside it are the chromosomes, or the genetic material. They have genetic material, DNA, which we'll talk about separately, the differences and similarities, but as far as the nuclear structure itself, there is no membrane surrounding it. That's also because it's not only that they lack a membrane, but there's no nuclear membrane, there's no other membranes at all. Okay, so no internal membranes. So no phospholipid bilayers inside the cell, creating endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, all these other sorts of structures that are common for eukaryotic cells. The prokaryotic cells don't have them, including the nuclear envelope, which that plus the DNA and proteins all make up the nucleus, or the nuclear structure. So there is no nucleus, but there are also no membranes. That's going to be common for the prokaryote groups that are the Eubacteria and the Archaea. They're both going to have that in common. So what I'm going to do is kind of break this down into sort of three parts, three separate lectures. There's going to be this one focusing on Eubacteria, their characteristics, another one then focusing on Archaea, their characteristics, and then a third one that will kind of be a compare and contrast between the two, because they'll have certain similarities and then certain things that are unique. And I'll point some of those out as we go along, but I'll then particularly highlight them in a specific future summary lecture as well. I'm also going to show you that I'm going to break down not only this lecture, but many of the future ones about anything, about prokaryotic cells, about plants, about fungi, and all these sorts of things into categories like these. So generally I'll be talking about the organism, whatever the organism happens to be, and its structure or unique structures that they have, referring to what's similar to other groups and then what is unique about them. And then we might go into some more detail with some of those structures, and others we might just kind of gloss over because there's not enough time to cover everything in detail. We'll talk about physiology, essentially how the organisms get nutrients and how they use the nutrients, what types of metabolism they have, variety of oxygen usage and not that sort of thing. We'll discuss for many of the groups of organisms. How they move, if they can move, what do they use to move, flagella or cilia, do they have musculature like in animals, then we'll get into types of muscles, all that. Reproduction, how they reproduce, cell division, unique types of cell division, sexual reproduction, asexual reproduction, that sort of thing, and then the roles of the organisms. That's just kind of setting this whole thing up, and that's what we're going to talk about right now for Eubacteria. So as far as structure goes, what do we have? Well, there is the cell membrane. And that's a phospholipid bilayer. Just like we see pretty much in our cells. Now, so that's not particularly unique. Outside of the phospholipid bilayer, we're going to find another layer. Come all the way around here. And this is going to be unique. This layer that's outside the phospholipid bilayer is the cell wall. Now that is unique. You may recall the term cell wall related to plants and plant cells. Plant cells do have a cell wall. The cell wall of a plant cell is very different, made up of different materials and structured in a very different way. So this is unique, a unique structure. The cell wall of bacteria, of the Eubacteria, is composed of a material referred to as peptidoglycan. So we're not going to really go that much into the details of that. That's more for another course in microbiology, or focusing a lot more on the bacterial structure specifically. But generally, it's chains of sugars. So there, let's pretend these are like little sugars in a chain. And the sugars are linked together like this in strips like that, chains essentially of sugars. And there's different types, N-acetylglutamic acid and acetylmuramic acid. You wouldn't have to know that here. And then that's the sugar part, that's the glycan part. And then they are joined together with peptide bridges. So there's some amino acids connected to some of these sugars, and then they connect to amino acids from other sugars, forming a bridge, a bond between peptides, between amino acids. And then that's what makes the peptidoglycan structure. That's unique for the bacteria. Okay, nothing else really has that. So all the members of this group have that, and or some variation of it. So there are some variations of it that they may have. One of those variations being really one of the main classification characteristics for the group. So how are Eubacteria classified into groups? Well, one of the major characteristics that we use to classify them is cell wall. And if it's very thick peptidoglycan or very thin peptidoglycan. The thick ones are referred to as gram positive. I'm just putting GM for gram positive, and the thin ones are gram negative. And that's referring to a specific type of biochemical test, which is a stain, where we stain the cells and try to wash the stain away. And if the stain remains and isn't washed away, it's because of the thick peptidoglycan layer, they're gram positive. If the stain washes away, then they have no color to them, and it's considered gram negative. And then if we were to look at them, we'd have to apply some different kind of stain then to look at those cells. So that has to do with essentially how many layers of these sugars are there. Is it just one or two layers, or are there 50, 100 layers, very, very thick? So the gram positive ones, very, very thick. The gram negative ones, very thin. They have other unique characteristics. The gram negative ones will have a second, so not all these will have this, but some of them have a second phospholipid bilayer outside. That's what the gram negative ones will have. So they have a super thin little layer here, but that makes it a little more rigidity to it, a protection, and other characteristics which we could talk about maybe later as we add in some other things for the structure. Keep something else in mind. These layers aren't exactly right up against one another, so there's actually a space. So if you have the phospholipid bilayer, here, and then you have the cell wall, here, there's a space in between the two layers. So in between, say, like, if we're looking at, let's say, this little bit right here that we're looking at is, this area kind of here blown up. We're looking at like a close-up sort of view of that. And then there's some, potentially outer layer here, maybe another phospholipid bilayer if it's gram negative, but there'll be other layers I'll talk about in just a second. The space is important. It's called a periplasm, which is separate here from the cytoplasm. Right, so cytoplasm is the liquid compartment inside the cell membrane, and all those materials that are within that liquid, so other structures. So some things that bacteria have that are not particularly unique are, say, ribosomes. So found in this cytoplasm, We have ribosomes in the cytoplasm of our cell. Plants do, fungi do, protist cells do, bacterial cells do. All cells pretty much have ribosomes, and the ribosome has two parts to it, a large and small subunit. That's pretty much the same. There are some variations and differences between them if you get really specific, but overall they have the same function and the same basic structure. Now, what we know about our ribosomes and how they work is really from the studying of bacterial ribosomes and how they work. So they're very, very, very similar in this particular case. So there's a cytoplasm, a fluid filled space. In that space there's DNA. So the DNA genome of the organism. Something else about the bacteria. And this is going to be something true for the other prokaryotes as well. So I'll add it over here. They have circular DNA. So when we say our chromosomes, we didn't really get into the detail of the molecular structure, of the chromosomes. But the chromosomes have typically ends to them. And one end might have a phosphate group, and the other end would have an OH, or what's called a hydroxyl group, at the end. DNA was supposed to be double-stranded, right? So we have two strands of DNA. They're opposite one another, like this, and then there's hydrogen bonds joining the strands together. So this is supposed to represent the hydrogen bonds, the little dashes there. But this is called linear DNA, meaning that there are open ends to it. What happens in the prokaryotic structures is they join. So this P attaches to this OH is attached to this P, and it forms a loop-like structure. And so that's what we have here, and there are unique things about that in terms of DNA replication. So how it works, how it's edited, and other sorts of things that you might get to in, again, another class, but here we're just kind of introducing. What are these cells? What's unique about them, and what's similar to other cells and other organisms? What structures do they have in common? So, cytoplasm, DNA, ribosomes to make proteins. That's the job of the ribosomes. Okay, then we have a cell membrane. What else is in the cytoplasm? Well, there's a whole lot of other things, right? In the cytoplasm is where we'll have a lot of enzymes to start to run the cell's physiology. We're going to still continue with structure. We're not done with it, but we're going to start adding in here. So for physiology, the cells are going to require nutrients. Where do they get these nutrients from? Well, plants. Yeah, well, not plants. Bacteria can be autotrophs like plants. So a photoautotroph means they can use light and carbon dioxide to make organic molecules like sugars, and then they can break those down and then use them themselves for energy molecules to make ATP. So generally, regardless of how you start, I'll talk about a couple others here, some other terms, the goal is to make ATP. And that ATP is going to be the same ATP that we have in our cells. Let's say ATP plants need in their cells. All cells, all living things are cells or are made up of cells. Cells require energy to be alive, to carry out all the chemical reactions that happen inside a cell. That energy, not always, but often comes from a chemical molecule. That molecule is ATP. The cells typically don't get ATP from the environment. They typically have to make the ATP. So cells take in other molecules that they could use to make ATP. That's the cellular physiology or cellular metabolism that we talk about. And there are different ways of starting off and kind of getting to that point, right? But for most living things, that's the way it's done. There are other ways of getting and releasing energy. That's going to kind of tie in here. So our cells, if you studied any of this ever before, and anything else, have mitochondria. And we talked about mitochondria previously when we talked about the evolution of eukaryotic cells and serial endosymbiosis. The mitochondria are the cells, or are the structures within our cells that are largely responsible for making most of the ATP that we have. Bacteria don't have mitochondria like we do. There are no other structures here in the cytoplasm. So how do they make ATP? Do they do it in a totally different way, or what? They obviously don't have mitochondria, so do they have an electron transport chain? The answer is yes, they do. They have their own electron transport chain. It's essentially the same thing that's in our mitochondria, because our mitochondria were, at one point, bacterial cells. That's kind of historically. So there are pumps that push protons or hydrogen ions from the cytoplasm into the space between the membrane and the cell wall, that periplasm. And so this space then builds up hydrogen ions, and then eventually they can diffuse back into the cytoplasm, and that releases energy. And that energy is what's directly used to make ATP. That's the way it works in our cells. That's how our mitochondria make ATP. That's how bacteria make ATP. Pretty much the same way. So you don't have to know all the details of that process. That's something you can study in cell biology class. All the electron transport chain, the proteins of the electron transport chain, the energy flowing between the different active transport pumps, and so forth. But what you should know here is that it exists, where it exists, that it is in the cell membrane of the bacterial cells, right? And that it does pretty much the same thing that happens in our cells. Okay? Again, everything's going to have sometimes slight, very specific, if you start to look at the molecular structures, differences, but if you look at them on the general, the big picture, pretty much the same sort of thing. So they could get energy from light, and they could get their carbon dioxide as the carbon source, or they could be photoheterotrophic, and still use light. That's the photo part. But they need some organic molecules to start off with. So part of being a heterotroph is you have to ingest organic molecules. You can't just get your carbon source from carbon dioxide. Okay, you're not an autotroph. Heterotrophs have to essentially feed. Right? And so bacteria, how are they going to feed? Well, a couple different ways. So physiology, how are they going to get their nutrients? A lot of it is through absorption and diffusion. So molecules will be passed across all these layers. So many times they'll have pores or openings in them that they can move things through. Some of them can phagocytize other structures, meaning that the bacterial cell, I'll have to look in a little bit about this in the serial endosymbiosis. And then here's something else, a particle or another cell. And then what we can have occur is that bacterial cell can essentially form a little pocket like this, where it's kind of surrounding this other structure, and then eventually consumes it, where it could be broken down. So they can take in nutrients in other sorts of ways, okay, by phagocytizing. And what are those nutrients? Well, those would be then the bacteria that are our chemotrophs. So they're not getting light, using light for energy. They're using chemical energy from the breakdown of other molecules. So they have to break down chemical molecules like, sugars, just as an example, and get electrons from them, and those use those electrons to power other processes in the cell to eventually make the ATP, which is the molecule they'll use to do all these other sorts of things. So the metabolism of our cells and bacterial cells, and in many essential ways, is the same. And it's actually going to be the same for plants, and it's the same for other things. Now, bacteria, though, can do things that plants do and that we do. So they have there's a whole variety of types of metabolic processes. In addition to that, something I want to point out, for a lot of these reactions to occur that I've been talking about, they require oxygen. So this is another aspect of the bacteria. and their metabolism or their physiology. So some of them are what are called aerobic. That means they require oxygen, like us. Our cells are aerobic. We have to have oxygen. If we don't have oxygen, our cells will die because we need the oxygen actually to make the ATP, and we need the ATP to power all the chemical reactions in the cell. Without the ATP, the cells can't do anything and they die. Without the oxygen, we can't make the ATP, so again, the cells will die. So same thing for some bacterial cells. They need oxygen. If they don't get it, they'll die. Other bacteria are anaerobic. They do not require oxygen. And in fact, there are a variety of different types of anaerobic bacteria. I'll talk about these a little bit more as we talk about the Archaea, because they have some extreme types of physiology. So anaerobes can, some not use oxygen, but tolerate it. So they could be in an environment with oxygen and be fine. They just don't use it. Others that are strict anaerobes, oxygen is toxic to them. So if they're exposed to oxygen, they die. So that goes back again. And those are current day modern bacteria that are anaerobic. They cannot tolerate oxygen. And again, that goes back to when we talked about the history of life, and that there were some organisms that were the first prokaryotes. They lived on the earth before there was photosynthesis that would give off oxygen. So the atmosphere had very low levels of oxygen in it, and those organisms would be anaerobic. And then as photosynthesis occurred, it produced oxygen, probably killed off many of them, and then some were able to adapt in other ways. So we still have that today, a lot of anaerobic, bacteria. And then there's things in between where bacteria can actually do both. They could be aerobic and anaerobic. They can kind of do one and they could do the other. So there's a lot of flexibility, in that. So kind of keep that in mind, that there is some flexibility in whether they require or don't require oxygen. It's unique for certain species. Certain species can do without it and others cannot. Now, what's another one of the things that the energy is needed for? Motility. And this topic will become important also as we move past the prokaryotes and start to go into the eukaryotes. So as we start to talk about protista, once we get there, that will be a few lectures away. One of the things we'll use as a characteristic for them is also motility. Some have amoeba, some have cilia, some have flagella. And so these sorts of structures, I'm going to compare and contrast them in the future as well to what we're talking about now. But right now, we'll say that some bacteria have flagella. So for motility or movement, we have a unique type of flagella that's going to be, because we'll see in the future, very different than eukaryotic flagella. So in our cells, some of them have flagella, some of them have cilia. If we look at the biochemical structure of flagella and cilia, they're exactly the same. There's just it's the length that's different. If we look at the biochemical structure of a bacterial flagella, it's completely different than ours and the way in which it moves. So we're not going to, again, go into all the details of that, but I'm going to give you the quick overview of it. So in the cell wall, embedded in here are specific proteins called motor proteins. And then within those motor proteins are these flagellin proteins. There's a certain kind of protein called flagellin. And then they build this long structure like this, the flagella. Now, as they're embedded in this motor protein, the motor protein will actually rotate, circular, and it will spin. And as the motor protein spins, it then causes the flagella itself to rotate like this and spin. Cilia and flagella both move back and forth like a windshield wiper. And then the long flagella, because they're so long, they tend to start to get a waving characteristic. But if you were to shorten them at the base, they're just moving back and forth. Whereas the bacterial flagella is spinning like this. Our flagella do not spin. but bacterial flagella spin. So keep that in mind. The mechanism in which they move is different. It's going to require ATP. Ours require ATP. All those kind of things are similar. Now, bacteria can have a single flagella like this on one end. They can have flagella on both ends. It's called bipolar flagella. They can have tufts of flagella sticking off them like this. So different bacteria can have all sorts of different numbers and locations of flagella. Some of them can cover the entire cell, where the whole cell is sort of covered with flagella. So those are going to be some differences. But the flagella themselves, the structure of it, is going to be the same. Again, some differences between the gram-positive and gram-negative flagella because of the thickness of that cell wall and how they're attached. But overall, a lot of similarities. Another structure that bacteria have that do help them attach to other things and sometimes move around as well are structures called fimbriae. Again, we don't have anything like this, these fimbriae. These fimbriae are these extensions, from the cell membrane, from the cell wall. They kind of stick out like this, and they kind of can cover the whole entire cell, and they're specifically used for attachment. Largely, this is attachment to other types of cells, but it could be cells of the same type, but since they're not multicellular, these are single-celled organisms. They don't form necessarily colonies or groups or multicellular structures. So it's usually for attaching a bacteria to your skin, that sort of thing. All right, so we kind of have covered, these are some of the basic structures. Now there's more. A lot of people just picture the bag, this is the last thing I'll talk about here, of the membrane, and like liquid, and then there's just like a couple things inside. And that's a really simplistic picture of it. It's actually very, very complicated inside. There can be other sorts of structures. Bacteria can actually concentrate material into granules. So there can actually be these like little structures that are concentrated chemicals of some particular type. All right, so let's call this a granule. One of the purposes of our organelles is that they can store unique chemicals, molecules, ions, or these sorts of things in a particular place, so it's not all just in the cytoplasm. We can have calcium ions in the endoplasmic reticulum and very few in the cytoplasm, as an example. Bacteria don't have those membranes, okay, so they don't have those compartments, so they can't quite do as many unique things. That means that everything they have is all free in the cytoplasm, and it creates a tremendous amount of osmotic pressure. That means that water is always moving into a bacterial cell, or it's trying to at least move into the bacterial cell, because generally all the things in that cytoplasm are part of the concentration of that cytoplasm, and it's typically more concentrated with solutes of things dissolved in the water than anything outside them. So water itself then is going to want to always move in to try and balance that out, and that's going to cause a lot of pressure. So that means that inside a bacterial cell, there's going to be pressure from water. So osmotic pressure pushing on the membrane. There's roughly the equivalent osmotic pressure as inside the cell as there is air pressure in a car tire. So the amount of air in a car tire, the pressure pushing on the tire itself to inflate it, is similar to the amount of pressure pushing outward on that cell membrane, which is made up of phospholipids, which are really held together through nonpolar associations, which means you could just really easily get just blasted apart. And that's the purpose of the cell wall, is to kind of keep it tight, keep it together so it doesn't lose integrity and become destroyed. antibiotics, some of them, work by damaging specifically the cell wall and weakening it. So when they weaken the cell wall, pop, the cell lyses and it can die. That's not all antibiotics do not work like that, but some of them work with that particular way because they're specific for the bacteria. So knowing things about these organisms helps us understand them and how they affect us, as well. Which we're going to try to get to ecological roles here, but I need to talk about reproduction first, which will be somewhat simple because we're not going to get into details right now. The division of a bacterial cell is a process called binary fission. It essentially is where one cell divides into two cells, and they're identical. So we can call them clones, essentially, of one another. Now, this is how not just they divide. So our cells undergo a division process like this, mitosis, that typically produces two cells that are identical, and they would be essentially sort of clones of one another. Different variations can lead to some differences in gene replication, might make them a little bit different, but generally they should be exactly the same. But when we have reproduction, we undergo meiosis, right? And remember, meiosis, the biggest emphasis of that whole process was uniqueness and diversity, to create genetic diversity. So crossing over to create genetic diversity, things like mutation that happens that can cause genetic diversity, and all types of assortment that can cause genetic diversity, all those sorts of things in the gametes. This isn't just how they make more cells, this is how they reproduce. So this bacteria now has just become two bacteria. And then those were its daughter cells, essentially. Then they become two more bacteria, and they become two more bacteria, and they're all identical. That's not just for multicellularity, that's how they reproduce, just through division, which essentially means they'd all be identical, to one another. So we might assume that then, all bacteria are the same, and there's no genetic diversity within them. There's other mechanisms, which we're not going to really have time to get into, but I'll mention it for a little brief second here, is what can happen is one bacteria who may have some unique genes in it, typically separate from the nucleus. So these are separate from the actual, they don't have a nucleus, sorry, just their chromosomal DNA. Separate from their chromosomal DNA, they have other genes. So you can have, in a bacterial cell, other pieces of DNA called plasmid DNA. Not all bacteria are going to have that, but they can. That plasmid DNA can be passed from this cell to this cell. And now, so they'll duplicate it, so they'll keep a piece of it themselves, not going to get rid of it completely, but now this cell will contain it. So now these two identical cells, the product of binary fission, are not the same because one of the two of them picked up unique genes from a different cell. And it has its own genes, and so now it's different than this cell, it's different than this cell. They're all different from one another. So that's just a simplistic summary. There's a lot more details to that and how the process actually occurs, which we're not going to study in this course. It's in microbiology to study that. But you get the sense that they're not just completely limited. There's a lot more stuff that's going on there. But generally, how they divide is just through a typical double their DNA, separate the cells. They have all kinds of unique mechanisms to do it, but the end result is going to be two identical cells. Lastly, ecological roles. So this one's a big one. Again, we're not going to go into all of it now because some of this is going to come up later in the course, especially toward the end as we talk about like nutrient cycling and things like that. And so that's one of those big things. Nutrient cycling in the environment. So there's decomposition, which means, a tree dies, an animal dies, bacteria start to break down all the tissues of that organism, break down the larger molecules into smaller molecules, which go into the soil, which then get absorbed by other organisms, taken up as nutrients or raw materials for them to construct their own cells and body structures with. And then trees grow and animals eat those plants and other trees, and then they grow, and that energy, those nutrients, those even individual atoms get like recycled. But it's because the bacteria and fungi, which we're going to break them down, right, from the larger molecules to smaller ones. They can do things like nitrogen fixation. So our atmosphere contains a lot of nitrogen. It's actually around 80% nitrogen, the air you're breathing. But we can't directly use that nitrogen, and most other organisms can't as well. They can't take it. Do we need nitrogen? Yeah, for a lot of things. The amino acids that make up the proteins, so proteins contain nitrogen. DNA and RNA, they contain nitrogen. So we need nitrogen for a lot of these core super important molecules that code our genetic information, that make all the structures that do everything in the cell, but we can't use the oxygen in the atmosphere directly for that. Something has to turn it into some other molecule. So nitrogen, so it's just two nitrogen atoms bonded together with a triple bond, nitrogen gas, can then be used by bacteria to form things like ammonia. And then ammonia can be turned into other molecules like nitrates and nitrites through other biochemical pathways and processes. But the fixation part is really just taking the nitrogen from the air and then turning it into a molecule like this, which then can then have access to other biochemical pathways to turn it into other sorts of molecules, like amino groups, amino acids or nitrates and other sorts of things. And there's a ton of other roles of cycles, like that we'll get into, but just keep in mind in the environment, they're very important for nutrients in the soil and even in the atmosphere. For living things, bacteria are part of the microbiome of many living organisms. We have bacteria that live on our skin and inside our bodies. We actually have more than 10 times more bacterial genes in us than human genes in our body. And those genes are being expressed from the bacteria, and they are making proteins for themselves to just carry out their own activities, but they're also making some that carry out activities for us. So some digestive processes, the production of certain vitamins and essential nutrients, we can't do these things. It's bacterial cells that do them for us, that live symbiotically with us. And if we remove them, we become ill or unable to absorb those nutrients. There have been a lot of studies that demonstrate the dramatic and drastic effects of removing the normal microbiome from a living organism and how much it suffers. And really organisms don't develop properly, they can't grow properly, they can't do a lot of their normal metabolism properly, because a lot of the metabolism that occurs is bacterial. That's actually inside us, and even for plants and a variety of other organisms. It's bacteria that are helping these organisms survive. So it's a whole other topic, but it's beneficial. The other thing that we think about is disease, but that's been historic. The only reason why people really studied bacteria historically is the diseases that bacteria cause. And that's it's still important, but they do a lot of other things that are very, very important to survival and life in general on the entire planet, and for your own individual life, the bacteria that live within you. So there are a lot of other things that bacteria do that are positive, that outweigh some of the things that a few of the pathogenic species do, that are negative as well. One last thing, how bacteria are classified, to come solely back to the beginning. Cell wall was one of the first things. We often use a cell shape sometimes is used. So they could be like rod-like and elongated, or round like spheres. But the other thing that is really used is genetic sequencing. So it's So this stands for SSU is the small subunit, and rRNA is ribosomal RNA. So remember I talked about there were ribosomes in the cytoplasm of the cell. Well, the ribosomes themselves have two parts. A ribosome has a large subunit and then a small subunit, like this. This one's referred to in bacteria as the 16S, and our cells it's referred to as the 18S. And so the genes The code for this small subunit, the DNA sequence of those, are what is used to classify bacteria and look at their relationships. It's one of the main things that's used to do it. So we do use certain structural, morphological characteristics to classify them into groups, but we also use certain molecular and genetic comparisons between them, and this is the most common one that is used. So we're going to revisit these again as we compare them to the Archaea, but know that we're talking about Eubacteria, which is one of the domains of life. Okay, so this is a domain. Okay. Generally, they are referred to, and historically referred to, as prokaryotes, cells without a nucleus. But we said extended that to mean also no internal membranes. They have the circular DNA. They have a cell wall made of peptidoglycan, all very unique characteristics and traits. They can, not all of them have flagella, but if they do have flagella, it's of a very unique structure, unlike ours. They divide directly through cell division through a process called binary fission to make clones, yet they can receive genetic information from other cells just by what's called horizontal transfer. So they can just pick up pieces to become genetically unique and different. And they have a whole variety of roles for the biology of other organisms. And so these other organisms can survive and become healthy in terms of nutrients in the environment to individual organisms and their absorption of nutrients as well. So that's the first part of Eubacteria. The next thing we'll go into is Archaea.